very short, few minutes of introduction. I'll pass it to you, Hannes.
Okay.
All right, sounds good. Hi everyone, welcome again. I'll just quickly share my screen. Welcome to the webinar ProtComposer that is developed by Hannah Stark and Bowen Jing. They're going to talk about designing proteins with spatial control. Very briefly about the agenda, this webinar is going to take about somewhere between 30 to 45 minutes. I'll do a really brief introduction to your co-host, SuperBio AI. I'm the CEO and co-founder. My name is Berke Kujak. Then we'll talk about ProtComposer. Hannes is going to talk about ProtComposer. Then we're going to spend some time for questions and answers, either to Hannes or myself, and we're going to spend a few minutes with closing remarks. So just very briefly about SuperBio, we're a company, a platform, a software platform that is looking to accelerate research with generative AI. Really what we're building is this intuitive and collaborative AI platform for biomedical R&D. We provide easy and instant access to best-in-class AI models that eliminates the need for biomedical researchers to build IT infrastructure from scratch. And not only do we serve models, but we really built a collaborative software around the models that we serve so you can onboard as a team and work together with your colleagues, your scientists, your data scientists under one workspace. Really, the platform comes with a lot of AI models, really easy to use interface, APIs, collaborative features to share your data sets, your results, a data hub to manage your data, as well as access public data. And finally, this bring your own tools capability that if you have a tool and you want to make it public, we can deploy it for you. Or if you have a tool and you want to use it within your team, we allow you to do that privately through the platform as well. These are just some of the hot use cases that gets the highest traction. You're going to see a lot of protein design, protein relevant tools and models on this slide. That's why we were really excited for Hannah to talk and do a webinar on protein design. But besides protein design, molecular docking, literature review, and single cell is also very, very relevant on our platform. This is what it looks like when you navigate. You have hundreds of different AI models. If you're interested in using one of them, you can either click on one of these tags that brings back all the models relevant to that tag. And let's say you want to use RF diffusion by Baker Lab, all you need to do is just click on the name. That takes you to the app page, upload your data, or use the SuperBio demo data, select some parameters at the bottom as step two. And if you don't know what these parameters are, we provide you really helpful tooltips, or you can send us an email bottom left here if you have any problems or any questions, and then you can click on Run on GPU. That takes you to the results page. It runs in a few minutes. Once it completes, we send you an email, come back and view your results. You can do so through here. You can download this to local. You can share this job with the organization, and you can leave some comments for everyone else on your team to see. Um, so I will stop there. I'll talk a bit more about it at the end. We have hundreds of different AI models, as I said, but without further ado, I would like to introduce Hannah Stark, who is the co-author of ProtComposer, to do the presentation on their research. Thank you so much for joining, Hannah.
Alrighty. Then I suppose I should try sharing my screen
Go ahead.
and share some slides. Uh,
And just a note for everyone, if you have any questions, please feel free to type it into the chat. Hannes, I'll let you know if someone types a question so you can answer during the presentation as well. Please
thanks.
feel free to mute yourselves. That's not to disrupt the presentation. And thanks again for joining. Please take it away, Hannes.
All right. Oh, no, that's not the right slide that we want. This is the slide that we want. Yeah, and that's the paper that we're talking about now, a prod composer. And the what we do there, I guess, is to generate some protein structures like what you're seeing rotating around on the screen already. And this was done mainly together with Bowen, my co-author, and then our supervisors, I guess, were Tommy Arash and Carsten Kreis. And it was done with them as our managers and supervisors at NVIDIA in an internship. Yeah, and we had the privilege to during this internship improve their logo a little bit with our protein or make turn it into something more protein inspired. And the our motivation for what we want to do here is basically that we look at what already exists in the landscape of protein structure design. And mainly, this is dominated by things like RF diffusion, which Berge just told you about, showed you an accessible interface for. And what we can do there, right, is to, of course, unconditionally generate some structures. But what we can also do is we can take an input protein, like a target protein, and then we can generate the rest or an additional protein that then hopefully binds to this input, to this target protein. Or what we can do is that we take a part of a protein and then we generate a scaffold around it that harbors this little motif and keeps this motif in the structure that you specified at the beginning. Yeah, and then you have these proteins. You can find out what the sequences are that correspond to these structures. You can then order the DNA for your sequences, express them, and then hopefully you actually have the protein that you designed, and it hopefully also binds to the target or harbors the motif as you imagined it in your design. And if we now draw an analogy to the image world, then this or these types of applications, they're similar to the in-painting task, right? You have your target given, your binding target given, and then you generate the rest of the image, so to say, by inpainting this additional protein that then binds to the target. Just like in images, you have maybe the crop of an image given, and then it generates you the rest of the image that harbors your motif, so to say, if you imagine this to be the motif of your protein, if we want to talk in that language. Okay, but then what is it that we can do currently with image models that we cannot do with proteins, right? Because in the image world, we have many of these additional control capabilities where we, for example, can condition on a semantic map and, right, you just tell your model, I want some sky to be over here. I want some lake or some water to be over here and some greenery to be over here. And then it generates you an image adhering to that conditioning. Yeah, and we have things like being able to condition, for example, on text and then generate images that correspond to that text. Or lastly, as an example, we can make these controlled edits where you have an input image and then you have also an input mask, and then you can combine this with some additional text prompt where you want to say that you want to replace the mask with whatever you put in your text. And now we argue that for proteins, this is actually also a very important and interesting, or it would be very interesting if we could, and useful if we could have similar levels of control. Because natural proteins or function, or the proteins that fulfill important functions in nature, they're not just like a binder or a stable scaffold. There are these compositional molecular machines that are made out of multiple domains that each have their own functions. Like, as example, we can maybe look at this um, these transport proteins here in the middle, and then we, for example, have kinesin, and then you have here the part or the domain of the protein that attaches to the cargo that is supposed to be transported. Then you have these connection, or I guess it's called stalk, these domains, or this domain that just connects this motor with uh, these transporters, or with the part where you attach your cargo or that binds to the cargo. And then you have these other protein domains that like are the things that actually attach to the microtubules in the cell and then walk along the cell. Maybe you've seen movies of this crazy this crazy function happening sometime. And or animated somewhere. Yeah, and so we have these different domains that are composed together to fulfill different functions. And it would be nice if we could also be, yeah, like specify that we want to design a protein where this part has that function, the other part has the other function, and we can, for example, say in 3D space, I want this area to be a membrane domain, I want this to be a calcium binding site, and then we just give this conditioning to our generative model, and then we generate a protein that actually adheres to that conditioning and fulfills it. And while this is a big vision and the final goal that we envision, here in this paper we just take a first step in that direction, where we focus on specifying the structural layout and the secondary structure of the structural layout that you want to specify. And the way that we do this is that you specify regions in 3D space that you want to be filled with protein matter, and you specify which which secondary structure type they should have. So should it be a sheet or a helix? And yeah, the way that we specify these layouts is via these ellipsoids. And then you give these, let's say, layout or 3D layout specifications to the generative model, and then it generates your protein that adheres to this specification. And now or if you have some specification like this, you can, right, generate new proteins conditioned on them and by hand specify what your desired layout is and generate a protein according to it. But what you can also do is, right, you maybe maybe you saw in at the beginning when Berke reviewed uh, like this thing or this protein generated with RF diffusion, and you literally just got a single helix out of it. And we know that these protein structure generative models, they have a strong bias toward generating only helices and toward generating proteins with that aren't very diverse and just helical bundles, and proteins that have low novelty compared to what already exists in the protein database and in our training data. So what we can then do with our generative model that is conditioned on these 3D structure layouts is that we can just have a very simple statistical model that cannot possibly be overfit on the 3D layouts that exist in the protein data bank. And then we draw some 3D layouts from that are novel and diverse, and then condition on those layouts, and then the generated proteins, if they adhere well to the layouts, then they will also necessarily be diverse and novel. Yeah, and we can address this lack of novelty and diversity problem a little. Okay, but then why do we specify the stuff as ellipsoids? And our argument is that this is a nice trade-off between a very fine-grained between very fine-grained specifications of what you want to generate or of a 3D layout and very coarse-grained global specifications. And in the middle of this, there are ellipsoids that are, yeah, maybe I should go to the next slide, that are the right trade-off between expressivity, like that you can still do what you want, and easy to construct, being easy to construct, so that you as a human are still able to specify it, right? If you, let's skip the cut ID stuff, but let's say you had a voxel map, then specifying that by hand, you as a user, that's basically like already specifying the structure of the protein. So yeah, I mean, then I wouldn't need a protein structure generative model if I, as a human, could by hand specify the voxels where I want to the protein to be. And so this very fine-grained representation will not work out. And but we still want to have a large level of control, and we think ellipsoids are a fine trade-off between that. And what's like let's just say we now want to train with these ellipsoids or go with this ellipsoid route. So the way that we train our model is that we then have our data from PDB, our protein structures. We extract the ellipsoids that correspond to that data. and then we take some protein structure generative model, and we right, okay, I should also say this will be a diffusion model, right? Um, and so to train this, we then feed as input to this diffusion model the noisy structure plus our conditioning information. And then we predict the clean structure from it and have some type of diffusion model flow matching loss. And the only thing that changes from the normal diffusion model training is that we feed these ellipsoid layouts as additional input to the diffusion model. And then the diffusion model can decide to either look at this additional information or it could completely ignore it, right? But um, if it's or it is very helpful information for minimizing your flow matching loss, right? Because it tells you already the 3D layout. So the model will start looking at it and will start using it to better minimize its generative modeling loss here. Okay. Um, I guess here we just have spelled out what I said. But what are these ellipsoid layouts actually? Well, they have a shape, right, and a position. And this we represent as a center, which we call mu, and the shape we specify as a covariance matrix. I know why is that? Well, you can imagine these to be like little Gaussians. Like each ellipsoid corresponds to a Gaussian. The Gaussian has a center and it has a covariance matrix. And then the ellipsoid is defined by the surface of equal probability or the surface with equal distance. Um, yeah, with some cutoff. And that is then the ellipsoid surface. Okay, but the ellipsoids actually contain also a secondary structure annotation, like a type. Is it yellow or blue here? Slash, is it secondary structure beta or alpha? And then it has a number of residues that belong to the ellipsoid. All right. But then how do we obtain such ellipsoids from, like, right, these ellipsoids from a protein? The first step is that we label the secondary structure with that we label the residues with their secondary structure. So for each residue, we use this program, DSSP, it's called, runs very fast, just can take a protein structure as input and then tells you for every residue is it helix or is it a sheet? And then we draw an edge between two residues if they are within a certain distance and if they have the same secondary structure type. Then we end up with these graphs here, right? Where we have some connected components. And then we take these connected components and say that each one of the connected components belongs to an ellipsoid. And then last we obtain the ellipsoid by fitting a Gaussian, like a 3D Gaussian, to the 3D point cloud of that connected component. Right? And then you have your mu and your sigma to have your shape. And of course, we also know what the secondary structure type is and the number of residues in that ellipsoid. Good. We now have learned how we can take protein structures and extract um, ellipsoids from them. So we can train our model with them, right? And here we then first have like a little visualization of what our model is. And so that's the original multi-flow model, and we need to feed the ellipsoid information into that model somehow. Right here, we just have the multi-flow model, which takes these frames of protein residues as input, and then gives you an update of these protein frame residues, or it predicts the clean protein, and then with that, you perform an update of your noisy structure during generation with a flow model. Yeah, and now the thing of how we feed in the ellipsoids is that we have these two components. We have additional ellipsoid tokens that are being processed, and we have this what we call invariant cross attention, which is an attention mechanism to these covariance matrices, which is like something that I guess doesn't exist yet. Good. How does this work, this invariant cross attention?
Hannes, I think there's a question on the chat, by the way.
Yep, yep. Um, would you mind reading it out? So,
Sure. So Burchin asks, how do you assume linker regions in terms of length and disorder?
okay. So we have this when we generate our maybe Sal, you could mute yourself again. Um, so we have these regions, or we have our ellipsoids, right? And then the in between, we also have some these loopy regions, which now just don't belong to any ellipsoid. So if you feed the ellipsoids to our model, let's go maybe. mm, Yeah, let's just look at this. If you feed the ellipsoids to our model and you always then train it with some protein structures where there's still loopy region that is not inside of any ellipsoid, well, then it will learn that usually your ellipsoids, they have to be connected by some loopy region, and it'll still put some residues there. And yeah, as long as you have enough budget, so to say, for additional residues that you can allocate to loopy regions, your model will put them into loopy regions to connect your ellipsoids. And basically, if you start generating with a certain number, let's say big N of residues, then the big N minus all the small n of your ellipsoids will be the number of residues that will be allocated towards loopy regions that connect the ellipsoids. Yeah, and now to how this ICA component works for conditioning, like for so what we want to achieve with this invariant cross attention is that each residue sees is informed by where are the ellipsoids in 3D space with respect to that residue and compared to the other residues. And the way this works is that so each frame, right, it has a rotation and a translation associated with it. Each residue has a frame and a rotation and translation associated with it. Then you also have these um, mu's and sigmas, which define our ellipsoids. And let's say you have k ellipsoids. And then we now want to update this residues, so the ith residues representation based on information coming from the ellipsoid. So we want to basically pass messages from all of our ellipsoids to this particular residue, to this frame that then tells the frame where are all the ellipsoids and what is the shape of all the ellipsoids with respect to my own orientation and positioning. Okay, and the way that we do this is, or right, similar to invariant point attention, we first put this ellipsoid center, the k ellipsoid center, into the frame of reference of this or into this frame, so to say, by translating it and rotating it according to the frame. Then right, because now you do this for every or this redefining of the coordinate system. You do that for every residue, and you just do it with respect to its frame. You can just feed that into a simple linear layer, and all these numbers will be embedded in an SE3 equivalent manner. And then we add this to the initial representation of the layer to construct our keys and values. And uh, then we actually also add like an embedding of the covariance matrix after rotating it into the frame of reference of our frame, or the coordinate system of reference of our frame. Oops. And yeah, then this is how or these are the queries, keys, and values that we feed into our uh, into some attention mechanism. And basically, you can see this. Um, Basically, then the decay indexing, right, of our ellipsoids. If we have k ellipsoids, you could see as attention heads. Okay, but that is just particular type of how we feed the input into our uh, protein structure generative network. And you could do this in different ways. We tried other ways, and well, this is the one that we found to work particularly well. But then once we've trained the model like this, let's lay out how the inference actually looks like, right? So one thing that we can do at inference time is, as we said, we can take an existing protein and extract ellipsoids from it, and then we can edit these ellipsoids or change them somehow to edit our protein. And we could also take ellipsoids that come from that are manually specified by hand, um, to like just achieve the 3D layout of a protein that we are interested in, that we want, or we can um, like use our generative model over 3D ellipsoid layouts to come up with the ellipsoid layouts. Then we feed this into as conditioning into the backbone model, the denoising model, and it then just denoises the noisy point cloud to obtain our final generated structure that hopefully adheres to the conditioning input. And this way, right, we have this inference process where we're using this conditional vector field defined uh, by our flow model. But then maybe you know from the image world the principle of classifier-free guidance, so to say. And we can now also apply guidance to our, or use this guidance principle for our flow model, where we have our conditional vector field that pays attention to the ellipsoids and tries to adhere to the 3D layout conditioning, and the original vector field that just generates highly designable proteins. So proteins that for which it is sure that if you uh, get a sequence for them, that this sequence will fold up into that structure, and that this structure actually, or this sequence actually corresponds to the structure that you generated. Yeah. And this is then a trade-off, right? If you have this model that strongly adheres to the ellipsoid conditioning, then it's maybe the ellipsoid conditioning, or right, if you just have an additional constraint that you need to optimize for, then you can't, or you will lose your other, or will pay a debt in this designability category. And now what we can do with guidance is to trade off between these two vector fields and decide do we want something very designable, or do we want something that very much adheres to our ellipsoids. And then what we integrate with, or the vector fields with which we update our noisy point cloud, is no longer just the conditional vector field, but it is this interpolation between the conditional vector field and the unconditional vector field. Yeah, and we visualize this here as well, where in the background, so first we have two rows here, and in each row you have a different ellipsoid conditioning. And then in each column, we draw a sample from our generative model. But in each column, we have a different guidance strength of how strongly should the generated structure adhere to the ellipsoid layout. Yeah, and then we find that, of course, if you have no guidance at all, then it doesn't adhere to the ellipsoid layout at all. And as we increase guidance, adherence to the ellipsoid layout becomes stronger. But maybe the designability of the structure also decreases, right? You can imagine that this helix bundle here is very designable, or this one, um, while these ones aren't as designable, but they actually are more natural looking, have the same or like a more plausible amount of loopiness and like a more compositional layout. And we can also then see this quantitatively if we look at some metrics of how well does our conditioning adhere to how well do our generated proteins adhere to the conditioning. And the, uh, these plots here, they show on the x-axis always this guidance strength, and on the y-axis, one type of metric of how well our proteins adhere to the conditioning or to the ellipsoids. And let's maybe look at the coverage metric here, which just means how or what fraction, so to say, of the volume of the ellipsoid is covered by generated protein matter. And yeah, we see that if we have no guidance, then this is worse than if we have some guidance. Yeah, but then, right, if we have the setup, what we can do already is you can just generate, specify some ellipsoids by hand, and then you can um, yeah, get a protein that adheres to your 3D layout specification. The other thing is you can take an existing protein and then redesign its connectivity by taking the ellipsoids from that protein and then regenerating the protein corresponding to these ellipsoids. And that's what we're visualizing here, right? What you have in the top row is always a protein from the protein data bank, and then the ellipsoids that we extract from this protein. And then what you have at the bottom is the thing that we generated, or the protein that we generated when conditioning on these ellipsoids. And you see that the proteins here look maybe kind of similar, but the TM score is very low between them because the TM score also takes the connectivity of the protein into account, right? So we can measure, or we can see that in all of these cases, which are just randomly picked, the connectivity of the proteins changes significantly. Yeah, but then one question is, how can we kind of generate something completely from scratch? And for that purpose, we use the simple statistical model that I talked about that generates, or that's a generative model over 3D ellipsoid layouts. Then once we have, or then a protein generation works in a two-step process. We first generate a 3D layout with your one generative model, and then you generate the protein conditioned on that 3D layout with our other generative model. And to do that, we have this simple model over 3D ellipsoid layouts, which is just a Gaussian over the centers of the ellipsoids. And then you have this parameter, which basically controls the spread of your ellipsoids, the sigma in your Gaussian. And then it is a Y-chart distribution over the covariance matrices over the ellipsoids. So a Y-chart distribution is just a distribution over covariance matrices. And it has these parameters with which you can control the size of the ellipsoids and their roundness, so to say, their anisotropy. And then the last term here is an energy penalty for ellipsoids if they overlap too much, or over here if they overlap too much, or if they're too far apart, which is just this Mahalanobis distance between the ellipsoids. And then the way that we sample this, right, is we sample from a Gaussian, we sample from a Y-chart, and then we put it into our energy function, evaluate the energy, and then do a rejection sampling with that energy. Yeah, and this way we have this simple statistical model over ellipsoids, right? Which is just has these few parameters, and we can see how we can change or vary these parameters to obtain different types of ellipsoids. And then let's look at how we use that in our experiments.
You have
Uh,
a few questions, by the way, Hannes. Would you like me to
yep,
read
yep.
to you? So the first one is how are the overlapping regions treated? I guess you've
The
answered that already.
the overlapping regions of ellipsoids. Maybe the person can follow up in chat and specify if for what situation or what situation they're referring to. Is it a concrete question of how how does this energy function here work, or was this a question about what happens if we have overlapping ellipsoids in the generative, like as input to the generative model? What will that cause?
Hannah said
Uh,
ellipsoids, yeah.
Sorry, say it again.
They said ellipsoids.
Okay, we're dealing with ellipsoids in both scenarios, but I'll now just assume that we're talking about the scenario, right, where we have some, let's say, some input to our generative model. Uh, ah, like this here, right, where we have some ellipsoids that overlap with each other. Um, Well, you could have the scenario in which you, for example, in which you have two ellipsoids that where one ellipsoid is completely within another ellipsoid, and this is still a completely valid ellipsoid layout. And one way or one situation in which you will have this is where are my beta barrels? Here, right? What will be the ellipsoid of this beta barrel? Well, it will be a full, like over here, it will be a full ellipsoid like this. But then in the middle, you can have another ellipsoid that corresponds to the alpha helix in the middle of it. And yeah, this other alpha helix is just um, completely within the other ellipsoid. And how will it be handled? Well, it's just an input to the model. And then the model will see at training time, usually if I have an ellipsoid here and an ellipsoid over there, and the ellipsoid is intersecting with this other ellipsoid, then that means that probably the smaller ellipsoid is the one that should take priority, so to say, and where the protein matter should go if this has it, and what secondary structure type it should have in this region compared to the other ellipsoid. Yeah, then what are some other questions? Let's go through them.
Sure. Hannah also asked, how is the guidance performed?
How's the guidance performed? Well, right, I now assume that the question is concretely about how we add these vector fields together. Right? Because we have in multiflow a vector field that corresponds to the rotations of your residues, to the translations of the residues, and um, you can call it a vector field or a generator for the discrete transitions that you make for your sequence, because you're jointly generating a sequence and a structure. And the way that this is done is we just add up the vector fields for the continuous quantities uh, multiplied with the right lambda weighting, right, for the translations and rotations. And for the discrete transitions, we construct a new probability distribution to sample the new discrete tokens or the new discrete residue types from. And this probability distribution is obtained by taking the logits and adding them up with the right weighting, which basically means like um, tilting the distribution between or interpolating between these two distributions or these two categorical distributions. And yeah, it corresponds to adding up the energies with their correct weighting, and the energies are just logits, are the points.
let's do one more and maybe you can move on to the final part. This is from Luis Pinto. Is there a way to specify the same ellipsoid layout for two helices and make sure that both helices diffuse to the same sequence?
No. Um, hmm, that's interesting. Uh, um, Maybe you can. It would be cool if you like I now assume that you have some application in mind where for which you would like this to be the case. And I will be curious what your application is. Maybe you can write it in the chat so we can look at it in the very end. I'll be quite curious. Good, but then let's finish off with some of these results. We've seen these here already, right? Just some crazy designs with some weird ellipsoid layout specifications. And then what else, or something else that we can do is, right, we can take a protein from PDB here in the column A and take its ellipsoids, and then we can edit its ellipsoids and then generate a new protein corresponding to the ellipsoids. And here, what we do, for example, is we rotate this ellipsoid a little bit, and then, yeah, indeed, in the generated structure, the helix is rotated a little bit. We can translate these sheets a bit, and then indeed, the sheets are further up than they were otherwise. We can change the ellipsoids, or we can invert their secondary structure type and do things like that. But maybe the main quantitative result is the trade-off that we can now achieve between designability and these other three quantities that we're interested in: the novelty and the diversity of our generations, and also how helical are they. And this we can look at by tracing out a Pareto frontier of under all sorts of different parameters of the model. What are the designabilities and the diversity slash helicity slash novelties that you achieve? So you would want, right, always very high designability and very high diversity. So something up here and or up here or up here. That would be the ideal scenario if you can generate something that's up here. But now we draw like 100 samples um, from each generative model and under a particular setting, and then you obtain 1 point on this graph, right? You have a distribution of 100 represented by 100 samples. And we do this for all sorts of different parameters and always draw 100 samples and then compute the designability, the average designability, and then the diversity of them. And then we end up with these Pareto frontiers for the different models. And we just see that with ProtComposer, the trade-off is um, rather nice. And I should also note that this black dot, I think that's maybe interesting for many. This black dot is what some random samples from the PDB achieve. So that's just to point out that designability certainly is not a perfect metric, right? You um, natural proteins, real functional good proteins, they're not 100% designable. Just to say that. And here we have a little visualization of basically some proteins drawn from the base model that is not conditioned on ellipsoids. And here's some proteins drawn from ProtComposer with ellipsoid conditioning. And what these visualizations show is that always a generated protein together with the ellipsoids that correspond to the generated protein, not the ones that were given as input to the model, right? This is just a visualization to show that basically using the ellipsoids as a visualization tool of how diverse the secondary structure is of the generated proteins. And you can see that basically the base model very often generates these single blobs of just alpha helices, whereas this is not so much the case if you use proteins and ellipsoid layouts from our ellipsoid statistical model and ProtComposer. Cool. But then this would be the end of that deal. And I would be happy to look at the chat some more and maybe look at this application. Um,
There is one more question on the chat. Have you looked at generating diverse linkers for a fixed scaffold? For example, CDR loops of antibody variable domains.
no, we didn't try anything with antibodies. Um, and what's the like? Is there any experiment that we have that would somehow relate to that? Um, I mean, right, we take these original or these natural protein layouts and then redesign their connectivity in some experiments and then look at the TM score. But this is not a functional characterization of what these, um, yeah, also, right, honestly, I don't know why you would want to do this with ellipsoids. If you want to keep a scaffold fixed, then just keep the scaffold fixed and in-paint the rest with um, with your usual protein structure generative model. So I think this type of application would already be well covered. Oh yeah, I don't know. Maybe you have some application in mind where you would want some additional freedom in the scaffold and yeah, don't actually want to specify the structure of the scaffold, but only vaguely specify the structure of the scaffold and then generate the linker between them. But then have we made some design proteins real already? Nope. We only have computational evaluations in this paper, and this will also stay the same. But yeah, we are now working on other things beyond ProtComposer, where we are also not using these types of generative, let's say, frame-based models anymore, where we have some big collaborations going on with academic biology groups and groups from industry, where we want to build on bolts and maybe you know bolts, this clone of AlphaFold 3, and are building a model which we are calling BoltzGen, with which we aim to tackle various tasks related to generating proteins that are useful in the real world and to help out all our academic biology collaborators and other collaborators. Yup. Then, should we call it a day here or any
I think
other?
let's take one last question um, on the chat from Abraham.
Abraham. Is there a size limit of the protein for ProtComposer? Can it handle fused protein design? I don't know what fused protein designs are. Maybe you can quickly clarify. Is there a size limit? In principle, no. But the model has been trained with a certain crop size. I think it's 256. And if you generate proteins that are larger than that, then you will run into less and less well-generated proteins. And you would need to train with a larger crop size. Um, I now like maybe you can clarify what fused proteins are, but um, to me, when I think of fused, then I think of some fused genes that were fused together into a single protein, and I'm thinking you're just bringing them up because of the size and like them usually being larger, and then yeah, my previous answer applies, right? If it's larger than 256, to have higher enzyme pathway efficiency. Yeah. Um, then if it's larger than 256 or whatever crop size you train with, then you might start running into worse and worse generations.
All right. If you have any more questions, anyone on the call, add me on LinkedIn, add Hannes on LinkedIn. Um, Hannes probably has his email out as his academic in Google Scholar or something. But thank you guys, everyone, so much for joining, and thank you, Hannes, for the presentation. Really, really appreciate it.
Yep, friends of the sun. Thanks for the many questions. It was pretty nice. Like truly lots of questions. I appreciate it.
Of course. Thanks, everyone. Have a great rest of your Tuesday. Hope to see you in the next one. Cheers.
See you around.